Good afternoon. Good afternoon. It's not very loud, is it? Yes. Good afternoon and welcome, and thank you for your interest in the story that I'm about to tell you about the development of Benicaros, which is something that's very close to my heart and that we've been working on for over a decade. And we're now at the point that we can bring the benefits of Benicaros to consumers, which is a very exciting point in the journey that has brought us here. My name is Ruud Albers. I'm co-founder of Nutrileads. I'm an immunologist by training, and I'm currently the CSO of Nutrileads. Nutrileads is an evidence-based health ingredient company. We develop health-improving ingredients for the food industry and for dietary supplements. And we do that with a fairly small team, highly educated, 50% has a PhD, and we do that since 2012. We are located in the Netherlands, in Food Valley, in Wageningen, and we hope to have a global impact. What we want to do as a company is we want to help people strengthen their health through nutrition. We develop ingredients with clinically substantiated health benefits, and we develop the production process and navigate the regulatory trajectories to bring them to the point that they are ready to be taken up into food products and dietary supplements. And we focus on natural compounds from plants. We unlock biologically active compounds that are present in plants and develop them to the point that they can be a functional ingredient in food products. We're working on a range of health ingredients. You see our portfolio over here. But today I'll be focusing entirely on Benicaros, which is our lead ingredient, which is currently ready to be commercialized. Benicaros focuses on supporting immune health. Now, I don't have to explain anyone that immune health is actually important for health, and consumers also got that message. Since I started in this area more than 25 years ago, every consumer survey that was done came back with the result that consumers were interested in immune health. It was either on position 1, 2, or 3, wherever you asked. Now, what has changed due to the COVID pandemic is that it is on number 1 everywhere. All consumers worldwide are looking for solutions to support their immune system, to increase their resistance against respiratory tract infections. What we've also seen more recently is an enormous increase in the number of new product launches that play in this area, that claim something about immune health. In fact, it's the largest category at the moment, the category with the most new product launches at the moment. And then if you look at these products, most of them contain either vitamin D, vitamin C, zinc, and that's it. It's very difficult to differentiate. And consumers are confused. And the reason why these vitamins and that mineral zinc are in these products are good reasons. They are on a list that you can make claims on the effects of these ingredients on the immune system. It's the textbook knowledge list of EFSA, which says that if you are taking these ingredients, that helps the normal functioning of your immune system. And that's true. There's a vast amount of evidence that if you are deficient in these ingredients, your immune system suffers and cannot function properly. So if you're deficient, it is important to supplement. But that's not all. Good nutrition for the immune system is not sufficient to get an optimally fit immune system. You also need to train the immune system. It's just like the human body. You need to feed it and you need the training. You need the exercise. And that's what we do with Benicaros. Now, we didn't start last year. We didn't even start 4 years ago. We didn't start 5 years ago. The work that led to the development of Benicaros actually started in 2006. You may ask, what took you so long? Well, there was a lot that needed to be sorted out. And there was a lot that we didn't understand at the time and that we start to understand now. In 2006, I was working at Unilever within research and development, and I was leading a research group that was focusing on improving health through nutrition, and we were asked to come up with an innovative health ingredient that could support the immune system and increase resistance to respiratory tract infection. So the first thing that we did is we looked around globally and with a team of about 25 people made a list of what's out there. And we came up with a list of vitamins, prebiotics, probiotics, herbal extracts, what have you. 470 candidate ingredients that we evaluated. And we evaluated the evidence that these leads indeed have an effect on the immune system in humans. So we focused on clinical trials. And we embarked on a series of in vitro experiments where we looked at the effects of a selection of these leads on various aspects of the immune system. And to cut a very long story short, we became very interested in a traditional Chinese medicinal extract from ginseng. It has been used for centuries all over Asia for various health benefits, but one of the more standardized extracts was actually developed to support immune function. And the reason we were interested is the fact that there were a number of clinical trials that had been published demonstrating that the immune system was enhanced after ingesting this standardized extract, and that the incidence and duration of respiratory tract infections was actually reduced. So we thought, great, let's use this in food products and in dietary supplements. And then we found that that was not possible. There were regulatory hurdles, sustainability issues, and cost was an issue. Ginseng takes 5 years to grow before you can harvest. So it's also very difficult to speed that up. So at that point, we embarked on a discovery project where we tried to identify the active compound in this standardized extract. So we embarked on activity-guided fractionation. We took the extract, made all sorts of different fractions, and looked for where the activity was. And we found that the active was actually a specific fiber that is not unique to ginseng, but can be obtained from food crops. And that was the major breakthrough, because that made it possible to actually apply it in food products and dietary supplements. And this fiber is called rhamnogalactorenan 1. RG1, I'll call it from now on for obvious reasons. And that is around 2011, 2012, and that was when Unilever decided to focus more on home and personal care, and I had the opportunity to actually acquire the patents, acquire the know-how, and founded Nutrileads with the sole purpose of bringing this discovery further towards the market. So what have we done since? Well, let me first go back to RG1. What is RG1? It's a specific soluble fiber that can be extracted from the plant cell wall, from various crops. It is a specific domain of pectins, and the trick is that you need to take it out of the matrix of the cell wall for it to be able to interact with the immune system and train the immune system to become more responsive. It's the hairy regions of pectins. It's the part of pectins that pectin producers want to get rid of because it doesn't contribute to the structuring purposes, but it has biological activity. And if you eat this RG1, either as a dietary supplement or as part of a food product, then it will survive passage through the stomach and it will signal to the immune system. There are specific receptors on the immune system that recognize patterns, and this is one of the patterns that's being recognized by the immune system. And this recognition leads to training of innate immune responsiveness. And then the remainder of RG1 will actually transit further down into your intestinal tract. It's not digested by the digestive enzymes, but it is fermented by the microbiota. And it's selectively preferred as a substrate by beneficial microorganisms and leads to production of metabolites that also have an impact on immune responsiveness. So it has a dual mode of action in influencing the immune system. And I have some evidence to back that up. We've done preclinical studies as Nutrileads where we took cells from the human immune system. We essentially took blood, isolated various cells that are important for immune responsiveness, and we added RG1 to them. And what we saw was that RG1 in the presence of these cells actually leads to increased responsiveness of these cells. Whether it's phagocytosis or production of cytokines, we do see this enhancement in the absence of microorganisms. So it's the parent compound that has this effect. And not only in vitro studies, we also did a clinical trial, a human proof of concept trial, in which we recruited volunteers, 3 groups of 20 volunteers that received either a placebo product, which happened to be an instant cappuccino powder, or they received an instant cappuccino powder that was fortified with RG1 in a lower dose and a higher dose. And they were given that for 4 weeks, and we measured the activity of an innate immune function at baseline and after 4 weeks. And we did see that dose-dependently we saw an enhancement of the responsiveness of the innate immune system. using this biomarker. And this was a biomarker that was also used in some of the ginseng studies. So it closed the loop to the original studies. And in those original studies, people had also looked at incidence and severity of respiratory tract infections. So for us, this was a very firm confirmation of our hypothesis that actually RG1 taken from a food crop could have a similar effect as that original ginseng extract. So yes, Benicaros does have an effect on the innate immune system. What does that mean? How does it work? Well, it means that if you ingest only 0.3 grams of Benicaros per day with your food, either as a supplement or as part of a functional food product, it will reach the small intestine, it will signal to the innate immune system, and it will increase its responsiveness. It will train the immune system. Then it travels further and it gets fermented by the microbiota. It will stimulate beneficial microorganisms, the usual suspects and a few more interesting ones. And it will also stimulate production of metabolites, short-chain fatty acids, and also other metabolites that are more specific to RG1. And these metabolites also have an effect on immune responsiveness. So this is in vitro. In the same clinical trial that I described before, if you look at the development of the microbiota at baseline or after 4 weeks in the high dose group, you see a stimulation of beneficial microorganisms. So how does it work? After Benicaros has signaled to the innate immune system in the upper part of the intestinal tract, it moves on, it gets fermented by the microbiota, stimulates beneficial microbes to proliferate, you get more of them, and stimulates them to actually produce metabolites that then have an effect also on the immune system. So this was what we needed to actually raise the funding to do the trial that I had been dreaming of for 10 years. While I was at Unilever, I've been in many discussions about how to measure beneficial effects on the immune system. What do you measure? Is there a gold standard biomarker? And the frustrating answer is there's none. And we have been in discussions with academic experts, with experts from industry, and also with experts from EFSA. And what we came up with in the end is the most convincing way to demonstrate a beneficial effect on the immune system related to increasing resistance to infections is to infect people. And that's what we did. So we then embarked on a trial with healthy volunteers where people were randomly assigned to a placebo group or a group that received Benicaros as a powder to be added to a breakfast item of choice. So very harsh conditions. Matrix effect do not matter. This was done for 8 weeks, and after 8 weeks, these people were exposed to a rhinovirus. That's a common cold virus in their nose. So we knew exactly on which day people were exposed to which virus, and they were all exposed to exactly the same amount. So we had a starting point there. And then following that infection, we monitored the development of symptoms, and we monitored the biological response of the immune system to that infection in 2 weeks after infection. And we did that using a questionnaire, a validated questionnaire for the symptoms. We used a nasal brush, and everyone today knows what a nasal brush is, so that's easy to explain, to collect some material in the nasal cavity where the infection actually occurs. And we used a nasal lavage, washing the nasal cavity to measure everything that was actually excreted into that compartment. Our main outcome was the clinical symptoms. And what you see here is the symptom score using that validated questionnaire, where you see the arrow indicating the moment of infection and the gray line indicating the development of symptoms in the people that received the placebo product. And after 4 days, you see that the symptoms peak, and after that, they start to decline. Now, the orange line is the line of symptom development in people that received Benicaros for 8 weeks before they were exposed to exactly the same virus, exactly the same amount of virus. And you see that the curve is lower and it's shifted towards the left. In fact, it's 20% lower, so there's a 20% reduction in severity of symptoms, and it's 28% shifted towards the left, so there's a 28% reduction in duration of the symptoms, which translates in 1.8 day less suffering from the symptoms of this experimentally induced infection as a model. So yes, we do have an impact on a real infection. The other thing that we did with these nasal brushes, we collected epithelial cells. Those are the cells that are being infected. Those are the cells that need to respond first. And we isolated RNA to measure the response to the viral infection. And what you see here is a checkerboard plot of that response. Each column is a subject. This is, in a case, it's a subgroup of 16 subjects. Each row is an interferon response gene. And the interferon response is the very important early response to all viral infections. If it lights up red, then there is an induction of a gene. This is the day before infection. This is the infection. This is the day after infection in the people that received the placebo product. This is. And you see in some of the people there is an induction of the genes, but not in all of them and not very much. And then one day later you see, day 6, you see induction of some genes. And only on day 9 you see a more widespread induction of this, what is supposed to be a very early response to a viral infection. And then by day 13 it's gone again. Now look at the group that received Benicaros before the infection. You see already on day 3 there's a much stronger induction of this early protective response to a viral infection, this interferon response. And the main limitation of this study is that we didn't sample before day 3, because this data suggests that actually the interesting part happens already before that. So we see a vast acceleration of the protective innate response to this virus. And we've measured other things, and I've just summarized that here in this cartoon, where you see the kinetics of various responses, and you see that all of these responses come up at some point. Some peak at day 6, most around day 9, and the symptoms already peak at day 4. So the response is a bit late to prevent the symptoms. Now this is what we saw in the people that received Benicaros 8 weeks for the 8 weeks prior to the infection. And you see that all of the curves have been shifted towards the left. All of the responses are faster. And you see the symptoms plotted as the orange area there, which are reduced in severity and reduced in duration. So how does Benicaros work? After it has signaled to the immune system in the upper part of the respiratory tract and trained the immune system, after it has been digested or fermented, not digested, fermented by the microbiota, stimulating beneficial microbes, stimulating production of metabolites that have an impact on the immune system, these cells of the immune system do their work. And they migrate through the body, after they've been trained in the gut, they migrate to other sites, including the respiratory tract, where they play a very important role in the early response against infections. And because they're trained, they respond faster. And because they respond faster, you suffer less from the symptoms of this experimental infection. So that's the science part. Now, in communication towards customers, as we do, or towards consumers, as some of you may want to do, you may not want to talk about symptoms or disease. Actually, you may not even be allowed to talk about disease. But you can also turn the curve around and say, well, actually, let's talk about immune vitality or immune fitness. Clearly what we see here is that this infection reduces immune fitness, or it reduces overall fitness. And clearly what we can see is that the impact on fitness is influenced by Benicaros. If you use Benicaros to train your immune system to respond faster, the impact of the infection is less. You have less problems with a clogged nose, less problems with a sore throat, less coughing. But there's also less impact on your ability to interact with other people, to walk the stairs, to exercise, to live your daily life. So also on quality of life attributes. And all of those effects were statistically significant. So many hooks for differentiating communication, to differentiate or add to communication that you can do based on the usual suspects: vitamin B, vitamin D, vitamin C, and zinc. So how do we produce Benicaros? Originally it was discovered from this traditional Chinese medicinal extract, which was not sustainable. Now we can produce Benicaros from carrots. In fact, we can produce it from carrot pomace, which is a side stream of carrot juice production. These carrots are grown in Germany by farmers that we know. We have a completely traceable supply chain, and after the juice has been pressed, the remaining pomace is used as the source material to extract this RG1, Benicaros, these unique soluble fibers that do signal to the immune system and that do have a beneficial effect on the microbiota and thereby train your immune system to become more responsive. Our final product is a white powder that can be added to supplements or to food products. So to sum it all up, Benicaros, after many years of research, and yeah, I'm sorry, I wish I could have done it faster, but it took this long. It's now available for commercialization. We do have the clinical evidence for differentiating communication. It's natural, it's plant-derived, it has GRAS status in the US. Our novel foods procedure is nearing completion, so that we can then also launch in Europe. It has a very broad applicability. And it has virtually no impact on taste or color or smell. It could look like something like this. This is what you get when scientists start thinking about products. These are mock-ups of products. But it could also be in your branded product, because we are B2B. We deliver the ingredient. We only ask from you to bring it to the consumers so that they can get the benefit of it. It fits with a clean label. It can be halal. It can be kosher if needs be. That's the story of Benicaros. 15 years in the making, launching today. Thank you for your attention. Any questions at this moment? If not, yeah?